Okay, so we've sort of been introduced to some various metrics for evaluating spatial model predictions. But and yet there's a diversity of metrics there, a diversity of different ways you can measure model quality given some data. But there's actually, that's kind of the start of our problems, right? Because the fact that you can calculate a certain type of model performance doesn't necessarily mean what you might want it to mean in the case of making these sort of spatial model predictions. And the reason is basically that the environment is spatially autocorrelated, and that's challenging to account for. And that the species distribution data is spatially autocorrelated in some cases because of the environment, or to some extent because of the environment, but to some extent because of phenomena that have little or nothing to do with those environmental predictors. And so the fact that we're using spatially autocorrelated data means things that are close together tend to be more similar than things that are further apart. So temperatures of grid cells that are close together tend to be more similar to each other than temperatures of grid cells that are further apart. We're just sort of speaking of broad averages here. But similarly, you're more likely to see an individual of your species near where you've seen other individuals of that species for a bunch of different reasons. And the fact that these two processes are spatially autocorrelated and we're trying to figure out how they are correlated between them just generates a lot of challenges. There's also, when it comes to measuring model predictions, there are other issues. So first off, one issue is heterogeneity. So let's say we're just trying to explain or predict the occurrence of this single data point in this geographic region based on this environmental variable. There's not a lot of variation in this environmental variable, so probably, even if we set the best threshold we possibly can, it's going to pick out this grid cell, but it's also going to pick out these two, right? And so almost any model you make here is only going to get the grid cell that has your species in it right 1/3 of the time. If it sets a hard threshold and says above, if you're this color for this variable, then you're definitely there, then the best you can do is getting it right 1/3 of the time. If you sort of increase the geographic extent, you pick up, because the environment is spatially autocorrelated, you pick up a lot of grid cells where the environmental conditions are quite different from where your species actually lives. And here you could probably build a model that, even if it's the same model as before, just evaluating it against this background will make this model look better. Because let's say we've got that same model that says that these 3 grid cells are suitable. Well, now it's saying 3 grid cells are suitable out of 25, and you've got your species in one of those. And that seems much better than, 3 out of 9. And you've got, and you're correct on one of those. Right? And so by picking up that extra level of heterogeneity, just by increasing our study area, we've increased our apparent model performance. We've picked up a lot of areas where it's really easy to say that falls outside of the kinds of environments that my species has been seen in. And the more we increase our study area and the more we increase the level of heterogeneity, the variance in our environmental predictors, the easier it gets, at least apparently, to distinguish where our species has been seen from where it hasn't. And so our performance metrics, particularly our discrimination metrics, tend to just go up and up and up as we add more and more study area, and particularly as we add more environments that are unlike the place where our species lives. And that doesn't mean that the model is wrong or fundamentally flawed. It means the way we're evaluating the model is affected by this environmental context. If you add a bunch of areas that are markedly unsuitable for your species, your model performance using discrimination metrics, or at least AUC, will go up. It's a really easy way to kind of game your models, which you should never do, to be clear. I'm basically saying this is a way that you can cheat. You can cheat this way on accident, but you can make a bad model look good just by adding in a bunch of really unsuitable habitat. So be aware when you're doing that. And this isn't just a question of geographic extent, right? We've got the same thing going on if we, had our species in the same geographic extent, but there was just less variance in the environmental predictor we're using. Here, it's going to be really hard to get a good performance metric from even the best possible model, and here it's going to be really, really easy. So this level of variation in our environmental predictors really affects, even if it didn't affect our models, which it probably does in most cases, it definitely affects our model evaluation, particularly using discrimination accuracy. So that's one issue. Another issue comes out of autocorrelation in the environment. So question is, what does an uninformative model look like? If we want to say our model is informative, we have to reject the hypothesis that it's uninformative. Now, when you're talking about a regular old linear regression, for instance, if you had a one-dimensional thing here and no spatial autocorrelation or anything like that, you would say an uninformative model is one that makes a prediction of average habitat suitability that does not change across the environment. So you've got a regression here with a slope of 0 and it explains nothing. It's just an intercept. So this is what an uninformative model means in a linear regression context. And in the context, if you were to transfer this idea to the species distribution modeling context, that would mean an uninformative model will be one that makes this prediction of the same suitability anywhere. So that's one way of looking at what an uninformative model does. Another way of looking at an uninformative model is to say, okay, well, maybe it's not making the same prediction everywhere, but it's just making random predictions. And so our prediction is just kind of all over the place. It's just kind of static, right? And so there's no correlation between our prediction and the actual suitability that we for our species, or there's no correlation between the environment and the prediction we make. So that's another way of thinking what an uninformative model would mean. And in the case of species distribution modeling, that would look something like this. Basically, no autocorrelation in the predictions you make, and so you've got possibly the same average prediction of habitat suitability here and here, but here you've got this absolute sort of noise. So there's two ways of thinking about what an uninformative model means. What does an actual uninformative model look like? Well, that's a really good question. I would say read this slide, pause here, see if you can do these steps on your own, and then visualize the model that comes out. So we're going to do this in exercises later, but just as a chance to sort of get your hands dirty and see if you can figure this process out, go ahead and pause here and try it. Okay, hopefully you've unpaused now and we're back. So what do you notice? Which of those null hypotheses actually, or ideas of what a null model would predict, actually makes a prediction that looks like your uninformative model from random point data? The answer is neither of them. You don't get something that looks like this. You do have spatial variation in the predictions your model makes, but it's not random. So in species distribution modeling, an uninformative model, a model built from garbage occurrence data, but with real environmental predictors, does not make a uniform prediction, nor does it make a random prediction. So when we're talking about a null expectation of the performance of a model, we can't have this in mind and we can't have this in mind, or we're just going to do it wrong. So yeah, this is my random model. I just took Monte, took random background points and started them in as presence points and built the model. And you can see this model says, hey, this part of Spain is actually substantially better than, Eastern Europe, Italy. I guess that's still Western Europe, but Eastern Western Europe. But it actually does a pretty good job of saying our species lives over here and not over there. The test AUC on this is not extremely high, but still, you will notice that this doesn't look like either of those two options. And so if this is what a model built on uninformative data looks like, our null distributions need to reflect that. So yeah, our environment is spatially autocorrelated, and our species distributions are spatially autocorrelated, and so that generates this potential for spurious correlations between our species distribution and the environment that actually are not in any sense causal. They're just related to the fact that we're correlating two spatially autocorrelated processes. People have done some decent work on making more appropriate null models. So one of the things that came out, it's a clever idea. I think there are some issues with it, but it's a really good point. Bond and McGill wrote this paper where they basically said, okay, you can build these species distribution models and get this high discrimination accuracy and all that sort of stuff. But that doesn't necessarily mean that the predictors mean anything to the species, which is absolutely valid. And so what they did is they said, okay, let's construct some fake rasters here. One that literally is just longitude, so this is, eastness, and one that is literally just latitude, just northness. And let's actually check our species distribution data up on here, build a model that essentially just has spatial autocorrelation in it with no environments, and see how well this does in predicting our species distribution. And if our empirical model doesn't do better than a model that's just based on northness and eastness, then maybe our empirical models, the processes we're inferring, aren't actually there. And this is a really, I think we'll talk about this when we start. We're going to do this in an exercise. I think this may be an overly strict condition, and we'll talk about why, but it's a really good point. We do need to be thinking in these terms. What does an uninformative, in this case, an uninformative set of predictors do? And how do our empirical models compare to that? Because if we can't reject a hypothesis that basically just says our explanatory power is strictly due to the fact that our species distribution data is spatially autocorrelated, if we can't reject that hypothesis, we have some thinking to do. Like I say, I think that this might be too strict, and I'll get to why when we sort of start doing the exercises. But there are other sort of extensions here too that actually try to construct a null distribution of expected performance of models based on uninformative data, like the exercise that we just did. And again, we're going to do this in depth in exercises, but just to give you an idea of how this works, we're going to talk broadly about Monte Carlo tests. So when we start doing the identity test and a range break test and the background test and all that stuff, those are all Monte Carlo tests. But these methods we're using here to essentially measure model fit are also Monte Carlo tests. So people often confuse Monte Carlo tests or Monte Carlo methods with Markov chain Monte Carlo methods, which are very prevalent in phylogenetics, and also there's MCMC GLMM if you want to do really sophisticated sort of linear modeling. So Markov chain Monte Carlo methods are a subset of Monte Carlo methods. So Monte Carlo methods, Monte Carlo is, if you're not familiar, it's a place where people go to gamble in Europe if they have a lot of money and possibly not a ton of sense. So Monte Carlo is a famous gambling town. And gambling is games of chance. So a Monte Carlo method is one that does stuff randomly over and over and over again to just sort of estimate the distribution of something. And so very broadly, anything where you see permutations or things like that, that is a Monte Carlo method. A bootstrap and a jackknife are both Monte Carlo methods, although those are more often used for estimating confidence intervals than anything else. It's still, you're randomizing something a whole bunch of times. That's a Monte Carlo method. And it's what's really interesting about this Monte Carlo methods, they were kind of an obvious idea going way, 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 way back. But they actually weren't really feasible in statistics until we started getting computers where we could actually do some complicated model 100 times or 1,000 times or 10,000 times, using nonsense in order to make these methods sort of tractable, right? You're not going to do this if every iteration takes 5 days, because then they get, a resolution on your p-values of 0.01. You're going to have to do this for a year and a half. So Monte Carlo methods are very much a modern statistical luxury. So be glad you got the ability to do this. We can do a bunch of things now that we couldn't do before just because our computers are fast. Okay. So I initially designed this lecture in terms of the background test and identity test and stuff. So that's what this this example is from. But the general idea of Monte Carlo test is essentially identical. So we've got two species here, purple frog and a red frog. We build species distribution models for them, and we measure some overlap between them. We have these metrics that we'll get to in a later lesson that say how similar these two distributions are. They give us one number that basically says how much do these two species distribution models overlap in their predictions of suitable habitat. So we've got an observation. And now we want to know, what does that observation mean? And so going back to the case of model evaluation, we could say we have an AUC score for our model, but we need to know what that means in terms of the expectation. So whether we're talking about niche overlap or model performance, what we usually want to know is, how does our observation in our empirical data compare to what we would expect if nothing interesting was going on? This is essentially the foundational idea of p-value statistics, estimating a distribution under the hypothesis that nothing interesting is happening. Specifically, nothing interesting is happening with respect to your alternative hypothesis. So estimate a distribution under the hypothesis that nothing interesting is happening. Compare your empirical observation to that distribution here. And ideally, you would like to say, if nothing interesting is happening, we would be very unlikely to make an observation like the one we make with our empirical data. So that rejects the null hypothesis, and we construe that as some level of support for the alternative hypothesis. There's philosophical and practical issues with doing statistics this way, but this is how frequentist statistics work, and most, if not all, these Monte Carlo tests are usually run this way too. Certainly the ones in the NM tools are. So this is easy when we have some analytical formula that allows us to construct this null distribution. So if we can just say, okay, if nothing important was going on, the mean of this statistic would be 0.2 and the standard deviation will be 0.1. If we could say something like that, then we could just say this is a normal distribution, and we could just look for the percentile or the quantile that our empirical observation is at in that normal distribution. This is literally what frequentist statistics is. It's just this sort of comparison where we have some analytical formula that allows us to solve for the probability density of this value in under the null hypothesis. But there's a lot of stuff where the expected distribution is unknown under the null hypothesis, or is just too complex to calculate, or whatever. And this is what we use Monte Carlo statistics for. So if we go in and we're talking about comparisons between species for overlaps here, we know those are bound from 0 to 1, but we don't know what the random expectation, the expectation is under the hypothesis that the points are random or whatever. We have to estimate that from the data, right? So we can't just analytically say, okay, the difference between these two species distribution models is expected to be normally distributed with such and such mean and variance. We have to actually figure it out ourselves. So, how do we get a distribution? Well, what we do is random nonsense over and over and over again. We randomize things in a way that makes us think, or that we think, is identical to what we would see under the null distribution. So, thinking in terms of, going back here a little bit, model performance, if this is our empirical AUC value, and we want to know the distribution of AUC values under the hypothesis that our data is uninformative, we need to estimate this distribution. And the way we do that, again, this is the distribution of expected AUCs under the hypothesis that our occurrence data is uninformative. The way we do that is we generate uninformative test data, right? So we do just like the exercise. We generate some uninformative test data. We build models. We measure AUCs. We do that over and over and over and over and over again, hundreds of times, sometimes thousands of times, and we see, okay, most of the time, if our distribution data is nonsense, most of the time we get a value around here. So we get an AUC of 0.1, which would be crazy low, but still. And then we can sort of take the relative density of values of AUCs here and say, you know what, we seem to be drawing from a distribution that looks something like this. And we can do this a whole bunch of times so that we can sort of measure this distribution very, very accurately. But essentially what we're doing is we're estimating that distribution of expected model performance under the null hypothesis that our data is uninformative or reflects no meaningful biological correlation with those, or not even necessarily biological, but no meaningful correlation with those predictor variables. And so by comparing the distribution under that null hypothesis to our empirical data, we can reject the hypothesis that our empirical data is uninformative, which gives us some level of confidence that our empirical model actually means something. So this Monte Carlo process is exactly this. It's basically doing something random a whole bunch of times to generate, it's not always a null distribution, but to generate a distribution that you can't solve for analytically. And in a hypothesis testing, we're typically comparing an empirical value to those distributions. The Monte Carlo methods are extremely cool because they allow you to generate statistical tests for things that nobody knows the math for. And in the process of doing that, once you sort of wrap your brain around it, the process of doing it can be incredibly easy. Like, it really often just boils down to hooking part of your process up to a random number generator and then seeing what comes out if you just crank the random number generator over and over and over again. It's not conceptually hard in that sense, but sometimes it can be quite difficult to appropriately figure out what a realization of that null expectation actually looks like. So is it randomizing the values of our predictor variables? Is it randomizing the location of our occurrence points? Is it doing both of those things? Sort of having some hopefully deep insight into what a specific randomization procedure means with respect to the distribution you're trying to estimate and how that relates to the truth you're trying to infer is actually, that's the challenge. The challenge is conceptual. It's really deeply understanding your alternative hypothesis and your null hypothesis, and then figuring out how to do some sort of randomization that reflects the thing you actually want to learn. Because you can do this in ways that are actually uninformative or informative about the wrong hypothesis. Yeah, so very cool process, often very easy to do in terms of the practical aspects of it, but they require thinking more deeply about what your hypotheses are and what your null distributions are than most people are typically used to.